I welcome you all to my YouTube channel. In today's class, I will be studying with pistil, Megasporangium and embryo sac. Okay. First of all, we'll be starting with Gynoshiam. Gynoshiam represents the female reproductive part of the flower. See friends, very beautifully you are going to observe the Gynoshiam. In between this androecium, there is a Gynoshiam. Okay. It is embedded within the thallus. Okay. This is the thalamus. Sorry, it is the thalamus. On this thalamus, there are arrangement of calyx first, corolla next, androecium next, and next whorl, the last whorl comes the Gynoshiam, which I am going to dissect it out. Okay. Yes. Here we can see Gynoshiam. This Gynoshiam is having the bulge part at the base, which is attached to the thalamus. Okay. Here this part is ovary. Okay. And this part is style and this tip is stigma. The pollen grain should come and sit on this stigma in order to reach the ovules in the ovary. See friends, don't be confused between carpel, ovules and ovary. One by one in today's class we will be studying detailedly about everything. Okay. Gynoshiam represents the female reproductive part of the flower. The gynoshiam may consist of a single pistil called monocarpellary, or many pistils, or more than one pistil is called as multicarpellary. Okay, you people understood? In case of monocarpellary means it is having stigma, style and ovary. Okay. Stigma, there are different shapes of stigma. Stigma might be linear or bilobed, flattened, etc. and all. Okay. If only one ovary, one style, one stigma is present, we are going to call it as monocarpellary. Okay. Multicarpellary means where the carpels are more in number. Within a flower, the carpels will be more in number. Okay. That condition, this is stigma, style and ovary. Stigma, style and ovary. So in that condition we are going to call it as a multicarpellary. Okay. Here further that, see, now we are going to study under multicarpellary. Okay. In case of multicarpellary only, when there are more number of pistils, if they are fused together, we are going to call them as syncarpus. In case of syncarpus, what happens? We can't differentiate how many carpels are there. Okay. Only by taking the section of this ovary only will be getting. Okay. Within one ovary, there might be more than one carpels. Okay. See, when we can't differentiate. Okay. In the hibiscus, what we are having here, ovary is fused, style is fused. In case of hibiscus, what happens? Stigma doesn't remain fused. Okay. The best example for syncarpus, where all the three, stigma, style and ovary fuses, is papaver. Okay. They have very beautifully shown in your book. Next, if the ovaries are fused, we are going to, if the carpels are fused, we are going to call it as syncarpus. If the carpels are not fused and they are multicarpellary in condition, we are going to call such condition as apocarpus. Each pistil has three parts: stigma, style and ovary. The stigma serves as a landing platform for the pollen grains. The style is elongated, slender part beneath the stigma. The basal bulged part of the pistil is ovary. Inside the ovary is the ovarian cavity called locule. Okay, the placenta is located inside the ovarian cavity. Recall the definition and types of the placentation that you have studied in previous class. Here inside we are going to see the first example of hibiscus. Here we are having ovary. This is thalamus. On the thalamus we are having ovary. Then we are coming across the style. Then we are having free stigma. Okay. Here style and ovary are fused. Stigma is not fused. Okay. It is incomplete fusion. Here it is a complete syncarpus ovary. syncarpous pistil in case of papaver. Here what we are having is stigma, style and ovary, all these things are fused, okay, and this is thalamus. Here it is the apocarpus. See, can you see here ovary, stigma and style and stigma. Ovary, style and stigma. All the carpels are not fused. We can count the carpels, okay. This is example of apocarpus ovary in case of Michelia. So when we are coming across this ovary, when we are taking the section of this ovary, we are going to see the structure of ovule in this fashion. Ovary. Now when you are going to take this section of this ovary, what happens? Within one ovary, when we are going to take the TS, there might be large number of ovules. It is irrespective of carpel. There might be only one ovule. Okay, it's not that one ovary should have only one ovule, one carpel should have only one ovule. Please friends, please don't get confusion between carpels and ovary and ovules. Okay, carpel, it means the number of stigma, style and ovary which are fused together, okay, all they are going to form called ovary. Large number of carpels together will be forming ovary. You people are getting? And this is a style and tip is stigma. You people got? Next, what happens in case of ovary? When we are going to take the section of the ovary, ovary is having a locule within it, a cavity within it, okay. This cavity we are going to call it as a locule. You people are getting? And it might be also having ovules. You people got the difference between ovary, carpels and ovules. And another thing, it's not that for one carpel only one ovule is present, or for one ovary only one ovule. There might be large number of ovules also, ranging from one to many. Now when we are taking, now we have understood that ovary, within ovary there might be countless n number of ovules, okay, from 1 to n number of ovules. So when we are taking only one ovule, and when we are taking the longitudinal section of this ovule, how it is going to appear is in this fashion, okay. This ovule is going to be attached for the ovary. See, here you can see ovule remains attached to this ovary. This is a ovary, ovules remains attached to this ovary by a funicle, a stalk like structure called funicle, okay. And the junction between this ovule and ovary, or the funicle, we are going to call that junction as helium, okay. This is outer integument, inner integument, middle integument of the ovary. This is the chalazal end and this is the micropylar end, because here micropyle is present. Through this only pollen tube can enter through this and through this way, okay. And at last we'll be having in the embryo sac, there will be having the synergids and egg cells, okay. In eggs, for the egg cell, the pollen tube is going to come here and it is going to fuse here, okay. In detail we will be studying all this thing in later class. Now we'll just be concentrating on the structure of the ovule. Now you all understood chalazal end, micropylar end, integuments and embryo sac, and this is nucellus. By eating the nucellus, with the nourishment of the nucellus, this embryo sac keeps on growing, okay. In completely matured ovule we can't find the nucellus, we can find only the embryo sac, okay. Now here we can call Microsporangia also for ovules. For ovules, see, Microsporangia we were calling for pollen grains. Please remember, Microsporangia, from Microsporangia microspores were coming. Yes or no? From microspores pollen grains were coming. So here for ovule we are going to call it as Microsporangia. Megasporangia. In case of Microsporangia we were finding the pollen sacs there. Here for the Megasporangia we can call ovule. The number of ovules in a ovary may be 1, to many. Okay, 1 in case of wheat, many in case of papaya, watermelon and orchids. Now we will be studying about ovule or microsporangia. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of stalk called funicle. The body of the ovule fuses with the funicle in the region of helium. The body of the ovule fuses with the funicle in the region of helium. Helium region, the funicle is fused and funicle is attached to the ovary. In this fashion, they remains attached. Thus, helium represents the junction between ovule and funicle. Each ovule has 1 or 2 protective envelopes called integuments, outer and inner integument. Okay, integument encircle the ovule except at the tip where a small opening called micropyle is present. See here, integument is not continuous. Here it is been ruptured or incomplete. Here this region we are going to call it as micropyle. Okay, organized opposite to the micropylar end is the chalazal end representing the basal part of the ovule. Okay. Enclosed within the integument is a mass of cells called nucellus. The cells of nucellus have abundant reserve food material. Located in the nucellus is the embryo sac or the female gametophyte. For the embryo sac, we are going to call it as female gametophyte. In case of male, for pollen grains, we are going to call male gametophyte. For female embryo sac, we are going to call female gametophyte. Okay. And that embryo sac, female gametophyte, is surrounded by what called nucellus. You people are getting, nucellus is the nourishing tissue of this embryo sac. And ovule generally has a single embryo sac formed from a megaspore through reduction division. Okay, meiosis occurs here and one megaspore is formed here. The process of formation of the megaspore from megaspore mother cell is called megasporogenesis. Even here reduction takes place. Ovules generally differentiate a single megaspore mother cell in the micropylar region. Most of the time at the micropylar region only the megaspore mother cell is going to differentiate and it is going to form for future embryo sac. This megaspore mother cell is a large cell containing dense cytoplasm and prominent nucleus. The megaspore mother cell undergoes meiotic division. After meiosis, 4 megaspores are formed, or among which only 1 megaspore is going to remain functional and other 3 are going to degenerate. Female gametophyte. In the majority of the flowering plants, one of the megaspore is functional while the other 3 degenerate. The only functional megaspore develops into female gametophyte. Okay, which is also called as embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. Okay, it means, we'll study here. See, at the micropylar end, what happens? There is nucellus. Okay, at the micropylar end, what happens? One of the megaspore mother cell is going to divide meiotically. After meiotic division, what happens? We will be getting 4 megaspores. Okay, firstly meiotic division will be getting 2, and the second meiotic division will be getting 1, 2, 3, 4. 4 megaspores will be calling them as megaspore tetrad. Among these 4, not all are going to be functional. Only one is going to be functional, and rest all all the 3 are going to degenerate. After degenerating, this degenerated megaspore is going to nourish the functional megaspore, and that functional megaspore is going to divide. See, now this functional megaspore is having only 1 nuclei. Okay, now rest 3 are going to degenerate. And see, now with respect to 1 nuclei, I am shifting for the another slide. And here 1 nuclei is going to divide and forms 2 nuclei. These 2 are again they are going to divide. This is going to divide and form 2. This is going to divide and form 2. Totally 4 nuclei are formed. Within one cell only the nuclei keeps on dividing. Now these 2 are going to divide and form 4, and here also 4. And these 4, now they are going to rearrange themselves. these three are going to rearranged at this part, chalazal end, and they are going to become two synergids and one egg cell. See, two synergid and one egg cell is going to be formed. Next, one is going to move towards the central end. See, it has moved here, and here also another moves towards the central end, and this two becomes polar nuclei. Okay. Next, remaining three are going to move to the opposite end or micropolar end. Okay. They are going to move towards the chalazal end, and these are going to move towards the micropolar end. Okay. These three are going to move towards the chalazal end, and they are going to become antipodals. When these antipodals at the chalazal end they are present, this chalazal end they are arranged in this fashion. Okay. See, at the chalazal end, three antipodal cells are arranged in this fashion, and at the micropolar end, two synergid cell and one egg cell is arranged, and at the central cell or the polar nuclei are present here in this fashion. So, wholly this embryo sac which is developed is only from one functional megaspore. See, all three have been degenerated and only one megaspore gave rise to this embryo sac. Means we are going to call such embryo sac as monosporic. Mono, only one, sporic, from one spore, one megaspore, this embryo sac has been developed. Okay. Now let us study the formation of the embryo sac in little more detail. The nuclei of the functional megaspore divides mitotically. This is megaspore. See, all three have degenerated. 4 megaspores were there, 3 have degenerated. One nuclei is going to divide mitotically, not meiotically. Mitotically they are going to divide and they are going to form two nuclei, and after formation of two nuclei they are going to move towards the opposite end. After the movement of the opposite end, the two nucleated embryo sac. Now we are going to call it as a two nucleated embryo sac. Two nucleated embryo sac. Okay. And now these two nuclei also are going to divide and they are going to form 4 nucleated embryo sac. Okay. Again next they are going to again divide and they are going to form 8 nucleated embryo sac. Now you can understand 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated embryo sac. So always what was happening after the mitotic division, there was the formation of the cell wall. Okay, here there is nothing such kind of formation of cell wall. Instead only the nuclei is going to divide here. Okay, now there is a typical female gametophyte or the embryo sac. Now there is arrangement of these 8 nuclei. 3 are going to move towards this side, and here at the micropolar end they are going to form synergid and one egg cell, and one nuclei is going to move towards the center, and this one nuclei is going to move towards the center, and another 3 antipodal cells are formed. These are synergids, this is egg cell, these are polar nuclei, and these are antipodal cells. Okay, in this form the 8 nucleated embryo sac is formed. Now you can count the number of nuclei: 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated embryo sac is formed. Okay. Now you can count the cell: 1, 2, 3, 4, 5, 6, 7. Okay, 8 nucleated, 7 celled, a typical angiospermic embryo sac is formed. Okay, in next class we'll be dealing with the pollination. Okay friends, thank you.